So an introduction to biomolecules. So before we can dive into looking at the specific biomolecules such as the carbohydrates, the lipids, and the proteins, we need to just have an overview of these biomolecules and the properties or characteristics that brings about how these molecules function. So our learning objectives will be for us to be able to describe the properties of carbon, the element carbon, and in its description, we need to be able to understand how it makes carbon a central component of organic compounds. How does carbon become central to organic compounds? And then we also need to define what an isomer is and be able to distinguish the three major types of isomers. And then the other objective is for us to understand the major functional groups that are present in organic compounds and also be able to describe their properties and what effects do they have on biomolecules or other organic molecules. And then we need to describe the relationship that exists between functional groups and the macromolecules. And then also be able to list and know the different kinds of biological macromolecules. And then should be able to understand also the relationship that exists between polymers and macromolecules. How do macromolecules build and how can they be broken down? So the outline for our lesson, first we are going to look at the element carbon and hydrocarbons. And then we are going to look at functional groups, isomers, and then we're also going to look at biomolecules as polymers. So we're going to look at biomolecules as polymers. So when you look at carbon, carbon is one of the most important elements, because you'll find that most of the biological molecules that will come across or that exist, they are predominantly made of carbon atoms that are bonded to other carbon atoms or to other atoms such as oxygen, hydrogen, nitrogen, sulfur, and phosphorus. So most of these biological molecules consist of carbon atoms. So when we look at a carbon atom, it has 4 valence electrons. So it simply means that for it to form its complete valence in the valence shell, it needs to form a total of 4 covalent bonds. So when you look at it, carbon can be able to form 4 covalent bonds. And due to that, we see that carbon is able to form straight chains and also branches. So carbon is able to form, of importance for the biomolecules, are the straight chains and the branches. However, carbon is also able to form rings, balls, tubes, and coils. So of importance for our course is that carbon is able to form straight chains and branches. So when we look at the hydrocarbons are simply molecules that consist of carbon and hydrogen only. So hydro for hydrogen and carbon from the element carbon. So hydrocarbon only consists of carbon and hydrogen. Now, those carbon hydrogen covalent bonds store a considerable amount of energy. So this is very important. The covalent bond that form between carbon and hydrogen store a considerable amount of energy. So you find that these hydrocarbons are usually used as fuel. So for example, you can give gasoline, which is a hydrocarbon, or which rather contains hydrocarbons. So it is rich in hydrocarbons. And also another example is propane gas. These are used as fuels because of the numerous hydrocarbons or because of the covalent bonds that exist between carbon and hydrogen. They are used as fuel. Now, as I mentioned earlier, carbon has the unique property of having 4 valence electrons. So it completes its valence shell by forming a total of 4 covalent bonds. So this is a very unique property of carbon, which ensures that it forms a backbone of the large and complex molecules that are essential to life. So each of the bond can link to another carbon atom or to another atom of a different element. For example, oxygen, nitrogen, phosphorus, and hydrogen. So when we look at the carbon to carbon bonds that are found in molecules, these are very strong and are not easily broken. However, it is not impossible for them to be broken. So we have molecules like our cells. We have cells. Our cells are able to break down molecules that contain carbon. And then also note that carbon does not only form single bonds. So carbon is able to form other types of bonds, able to form double bonds, in which 2 electron pairs are shared, which leads to the formation of a double bond. This is just showing a double bond between 2 carbon atoms, 2 carbon atoms. So we have double bonds. Carbon is also able to form triple bonds. It's also able to form triple bonds. Now, when we look at the single bond, the single bonds that are formed between carbon atoms, there is a degree of freedom of rotation on the single bonds. So single bonds are able to freely rotate. So this property permits organic molecules to be flexible, and henceforth they are able to assume a variety of shapes. So depending on the extent to which that single bond is rotated. However, when we look at the double and triple bonds, they do not allow rotation. So you find that in a region where a molecule has a triple bond or a double bond, it would tend to be not flexible. So it's going to be inflexible. So when you look at your chemistry of bonds, for the double bond, it's going to contain a sigma bond, what they call a sigma bond, and also a pi bond. So a sigma bond and a pi bond. So when you look at the sigma bond that is found in a double bond, for example, that bond is formed from hybridized orbitals, which are called the sp2 orbitals. So what happens that these orbitals can overlap to form one sigma bond. And so they form that bond between those two carbon bonds. This is also typical of the same bond that is formed in a single bonds between one carbon atom and another carbon atom. However, a second bond can be formed between the carbon atoms, which is called the pi bond. So the pi bonds are simply formed by overlapping of two spare p orbitals. So we find that these pi bonds do not allow a free rotation of the molecule. So they break easily and are weak. So they tend to resist rotation. So no wonder the double and triple bonds resist rotation. Unique to biomolecules are functional groups. So when we look at functional groups, they change the property of molecules. So they are going to account for differences in molecular properties. Remember, we looked at hydrocarbons. Now, those hydrocarbons are non-polar because of those because of the covalent bonds that form between hydrogen and carbon. So they lack distinct charged regions that are able to react with water. So hydrocarbons tend to be insoluble in water. So they tend to cluster and form hydrophobic interaction. Now, those hydrocarbons can be changed by the addition of what are called functional groups. So an atom of hydrogen can be replaced by a reactive group or a functional group that will change the way a chemical or compound behaves. So the properties of the major classes of biologically important organic compounds, such as the carbohydrates, the lipids, the proteins, and the nucleic acids, are largely a consequence of the types and arrangement of functional groups that they contain. So if we know the functional groups that a compound contains, this is very important. If we know the functional group that a compound contains, we are able to predict and know how it's going to behave. So we are able to predict its properties, its chemical, and also physical properties. So like I mentioned, just by replacing one of the hydrogen with one or more functional groups, the characteristics of an organic molecule is able to be changed. So these functional groups will determine the chemical reactions and associations that a particular organic molecule is going to participate in. Okay, so we have different molecules. We have different functional groups, rather. We have different functional groups. So let's look at the hydroxyl group there. One of the functional groups is the hydroxyl group. which is abbreviated as R connected to OH. R connected to OH. So the R is simply represents the remainder of the molecule of which each functional group is a part. So when you look at this R, simply represents the remainder of the molecule attached to the OH or the hydroxyl group in this case. So the hydroxyl group. So I said that when you look at the hydrocarbon, if one of the hydrogen atoms is replaced by a functional group, for example, hydrogen, its chemical properties are going to change. So for example, an example of a molecule with a hydroxyl group is an alcohol. So let's take an example for ethanol. And look at the example that is given there. It's ethanol. An example of an alcohol is ethanol. Now we have an example of ethane, which is a hydrocarbon. When we look at ethane, a hydrocarbon, it is a gas at room temperature. Ethane is a gas at room temperature. But when one of the hydrogen of ethane is replaced by a hydroxyl group, hence having ethanol or an alcohol called ethanol or ethyl alcohol, we find that alcohol, this ethanol, has cohesive forces because of the polar hydroxyl group that has been added. So you find that this ethanol will be a liquid at room temperature. Remember, ethane is a gas at room temperature, but we find that ethanol is a liquid at room temperature. This ethyl alcohol dissolves in water because of the hydroxyl group that it has. So it's able to interact with water. So it's highly polar. So it makes the compound more soluble through hydrogen bonding with water. And it may also act as a weak acid and drop a proton. So we have an example as hydroxyl group. We have an example of a hydroxyl group. So a hydroxyl group is very important. So of importance in biomolecules, we find hydroxyl groups in carbohydrates. We find hydroxyl groups in proteins. We find hydroxyl groups in nucleic acids. Remember, we already looked at nucleic acid and we saw hydroxyl groups. And one of the distinctive characteristic of RNA from DNA is the presence of the hydroxyl group on carbon 2 of the sugar. So we find hydroxyl groups in the carbohydrates, the proteins, the nucleic acids, and also the lipids. So this is very important. And then the other functional group that we have is a carbonyl group. A carbonyl group. A carbonyl group. So when we look at the carbonyl group will consist of a carbon atom that has a double covalent bond with an oxygen atom. So you have a carbon atom with double covalent bond to an oxygen atom. So this bond is actually polar because of this highly electronegative oxygen atom. So this carbonyl group is actually hydrophilic in nature. Now, the position of this carbonyl group is going to determine the class to which a molecule belongs. So the position of this carbonyl group is going to determine which class of molecule it belongs. So for example, we have aldehydes and ketones as examples of carbonyl groups. So carbonyl groups are found on aldehydes and on ketones. When we look at an aldehyde, the aldehyde has a carbonyl group positioned at the end of the carbon chain. So an aldehyde has a group positioned at the end of the carbon chain. While when you look at the ketone, it has an internal carbonyl group. So it's found between other atoms, between other carbon skeleton and another carbon skeleton in between. So it has an internal carbonyl group. So aldehyde, the carbonyl group is at the end of the carbon skeleton. All ketones have an internal carbonyl group. So we have examples such as from aldehyde, acetaldehyde, and also acetone as examples of carbonyl molecules or molecules with the carbonyl group. So the carbonyl group is found in carbohydrates and nucleic acids. So we're able to find carbonyl groups in carbohydrates and nucleic acids. Then the other group that is important for biomolecules is the carboxyl group. The carboxyl group, which is RCOOH. RCOOH. So molecules that can contain the carboxyl group are carboxylic acids. We have an example of carboxylic acids are able to contain a carboxyl group. So the carboxyl group, in its non-ionized form, consists of a carbon that is joined by a double covalent bond. So you have a carbon that is joined by a double covalent bond to an oxygen atom, as we have seen there, and by a single covalent bond to another oxygen atom, which in turn is joined to a hydrogen atom. So you have a carbon skeleton, we have carbon joined covalently by double bonds to oxygen, and also that carbon is joined to oxygen, another oxygen atom by a single covalent bond, and that oxygen is joined to a hydrogen. That's a carboxyl group, a carboxyl group. So carboxyl groups are important as they can be found in molecules such as proteins and lipids. Proteins and lipids will contain carboxyl groups. The other group that we have is an amino group. An amino group. So when you look at an amino group, which is abbreviated as RNH2. So the R is a carbon skeleton. An example of a family of molecules are amines. Amines. So when it is in its non-ionized form, like the example on the table there, in its non-ionized form, we find that it includes a nitrogen atom that is covalently bonded to two hydrogen atoms. So when you look at amino groups, they are weakly basic. They are weakly basic, because they are able to accept a hydrogen ion or a protein. They are able to accept a hydrogen ion or a proton. So they are basic. So amino group is basic. The carboxyl group, when we look at the carboxyl group, it is acidic. Now the carboxyl group and the amino group, functional groups, hence tend to attract or drop a proton. The amino group is going to attract a proton. The carboxyl group is going to drop a proton. Hence, the amino group function as bases, or the carboxyl group are going to function as acids. The most important types of amino and carboxyl containing molecules are the amino acids. Now, when we are going to look at proteins, we are going to realize that amino acids contain both an amino group and a carboxyl group. Amino acids contain both amino group and a carboxyl group. So amino groups are important components of amino acids and nucleic acids. So we're going to find an amino group in nucleic acids and also in amino acid. And then the other functional group is the phosphate group. The phosphate group. So a phosphate group is weakly acidic. It is weakly acidic. So I find that there could be an attraction of electrons by the oxygen atoms, which will lead to a release of 1 or 2 hydrogen ions, and henceforth it's going to produce an ionized form with 1 or 2 units of negative charge. So we know the phosphate group is important in, or is found in, biomolecules of nucleic acids. It is found in biomolecules of nucleic acid and also in certain lipids, the phospholipids. Remember that the phosphate group, the presence of the phosphate group in DNA is responsible for the acidic nature of DNA. So DNA is acidic due to its numerous phosphate group that it contains. So phosphate groups are important constituents of nucleic acids and also phospholipids, nucleic acids and phospholipids. Then the other important functional group is a the sulfhydryl group, which is R, the carbon skeleton joined to sulfur, and then sulfur joined to hydrogen. So an example of a family of molecules is thiols. So the amino, the sulfhydryl group is important as it is found in proteins. So amino acids contain certain amino acids that can contain a sulfhydryl group, which makes an important contribution to the protein structure. So for example, this functional group is found in cysteine. This functional group is found in cysteine. So you need to know these functional groups and which biomolecules contain these functional groups, which biomolecules contain the functional groups. So for example, which functional groups are found in carbohydrates? Which functional groups are found in carbohydrates? So we've mentioned that in the carbohydrates, we're able to find the hydroxyl group. In carbohydrates, we are able to find the carbonyl group, which could be a ketone or an aldehyde group. So need to know the functional groups in relation to the bio molecules. However, the functional groups are not the only responsible groups or characteristics that gives unique properties to the biomolecules. So we have the property of isomerism. So organic molecules can actually exist in different forms. So when you look at an isomer or isomers, these are simply compounds or organic molecules that have the same molecular or empirical formula, but have different structures. And hence, when you look at isomers, even though they have the same molecular formula, they have different properties. They exhibit different properties. So there are basically three types of isomers. There are basically three types of isomers, principally. So we have what are called structural isomers, and then we have what are called geometric isomers and stereoisomers. And of importance under stereoisomers are isomers that are called enantiomers. So three types of isomers: the structural isomers, geometric isomers, stereoisomers. And under stereoisomers of importance are enantiomers. So now when we look at structural isomers are compounds that differ in the covalent arrangement of their atoms. They differ in their covalent arrangement of atoms. So an example of structural isomers is glucose and fructose. So glucose and fructose both have the formula, molecular formula is C6H12O6, but they differ in the way their atoms are arranged. The covalent arrangement they differ in that, but they have the same molecular formula. So note that glucose and fructose are examples of structural isomers.